DNA is a double helix, which is then wrapped around histones, which are grouped together into what we call supercoils. Supercoils are what make up the chromatin, which is what chromosomes are made of. When untwisted and straightened, one can see what really makes up DNA. DNA is made of nucleotides, which are made up of phosphate, deoxyribose sugar, and a nitrogen base. There are 4 different nitrogen bases, which can all be paired with deoxyribose sugar and a phosphate to form a nucleotide. The 4 nitrogen bases are thymine, adenine, guanine, and cytosine.
Transcription is the process in which one half of a DNA strand is copied into a single helix strand called messenger RNA. This begins with an enzyme called RNA polymerase binding to the DNA and separating it. This process is initiated by a specific order of nucleotides called a promoter that varies depending on the protein that will eventually be created. Once the DNA is separated, RNA polymerase uses a strand of DNA as a template to construct the correct nucleotides. The mRNA nucleotides are identical to DNA, except thymine is replaced with a very similar nucleotide called uracil. Once all the DNA has been translated into mRNA, another sequence of nucleotides called the terminator instructs RNA polymerase to close the DNA strand and move on to make more RNA on other.
Translation is when the bases of nucleotides form the sequence that determines in which order to join together amino acids to produce a polypeptide chain protein. However, they need a ribosome to put them to use. Before the process can begin, the messenger RNA must be transcribed from DNA as well as released into the cytoplasm. When the messenger RNA attaches to the ribosome, codons move through the messenger RNA, which then codes for the amino acid to be made that corresponds to the codons. While this may be amazing, each amino acid is only carried by one transfer RNA molecule. After this process is complete and the ribosome has reached the stop codon on the messenger RNA molecule, it releases the protein, which completes this sequence of translation.
Protein folding. Once transcription and translation have been completed, the amino acids must be folded into proteins. There are 4 stages to this process. In the first stage, known as primary, the amino acids are configured in a curved chain, with each amino acid only connected to those directly next to it. In the next part of the process, called the secondary stage, either an alpha helix shown on the right or a beta pleated sheet shown on the left is formed due to the hydrogen bonding between the amino acids. In the tertiary, or third stage, 2 types of amino acids become visible. Hydrophilic, the amino acids that like water, and hydrophobic, amino acids that do not like water. In this stage, a chain becomes more three-dimensional and less like a chain, as the hydrophobic amino acids move towards the inside of the grouping, and hydrophilic move outwards, creating a sort of twisted blob. Finally, in the quaternary stage, this jumble of amino acids joins another to form a protein. This is BBC News.